Hemoglobin, also spelled hemoglobin and abbreviated HB or HGB, is the iron-containing oxygen transport metalloprotein in the red blood cells of all vertebrates as well as the tissues of some invertebrates. Hemoglobin in the blood carries oxygen from the respiratory organs to the rest of the body. In mammals, the protein makes up about 96% of the red blood cells' dry content, and around 35% of the total content. Hemoglobin has an oxygen binding capacity of 1.34 ml O2 per gram, which increases the total blood oxygen capacity 70-fold compared to dissolved oxygen in blood. The mammalian hemoglobin molecule can bind up to 4 oxygen molecules. Hemoglobin is involved in the transport of other gases. It carries some of the body's respiratory carbon dioxide as carbaminohemoglobin, in which CO2 is bound to the globin protein. The molecule also carries the important regulatory molecule nitric oxide bound to a globin protein thiol group, releasing it at the same time as oxygen. Hemoglobin is also found outside red blood cells and their progenitor lines. Other cells that contain hemoglobin include the A9 dopaminergic neurons in the substantia nigra, macrophages, alveolar cells, and mesangial cells in the kidney. In these tissues, hemoglobin has a non-oxygen carrying function as an antioxidant and a regulator of iron metabolism. Hemoglobin and hemoglobin-like molecules are also found in many invertebrates, fungi, and plants. In these organisms, hemoglobins may carry oxygen, or they may act to transport and regulate other things such as carbon dioxide, nitric oxide, hydrogen sulfide and sulfide. A variant of the molecule, called leg hemoglobin, is used to scavenge oxygen away from anaerobic systems, such as the nitrogen-fixing nodules of leguminous plants, before the oxygen can poison the system. Research history. In 1825 J.F. Engelhard discovered that the ratio of theta protein is identical in the hemoglobins of several species. From the known atomic mass of iron he calculated the molecular mass of hemoglobin to n times 16,000, the first determination of a proton's molecular mass. This hasty conclusion drew a lot of ridicule at the time from scientists who could not believe that any molecule could be that big. Gilbert Smith's Adair confirmed Engelhard's results in 1925 by measuring the osmotic pressure of hemoglobin solutions. The oxygen-carrying protein hemoglobin was discovered by Hunefeld in 1840. In 1851, German physiologist Otto Funke published a series of articles in which he described growing hemoglobin crystals by successively diluting red blood cells with a solvent such as pure water, alcohol or ether, followed by slow evaporation of the solvent from the resulting protein solution. Hemoglobin's reversible oxygenation was described a few years later by Felix Hoppe-Seyler. In 1959, Max Perutz determined the molecular structure of myoglobin by X-ray crystallography. This work resulted in his sharing with John Kendrew the 1962 Nobel Prize in Chemistry. The role of hemoglobin in the blood was elucidated by French physiologist Claude Bernard. The name hemoglobin is derived from the words heme and globin, reflecting the fact that each subunit of hemoglobin is a globular protein with an embedded heme group. Each heme group contains one iron atom that can bind one oxygen molecule through ion-induced dipole forces. The most common type of hemoglobin in mammals contains 4 such subunits. Genetics. Hemoglobin consists of protein subunits, and these proteins, in turn, are folded chains of a large number of different amino acids called polypeptides. The amino acid sequence of any polypeptide created by a cell is in turn determined by the stretches of DNA called genes. In all proteins, it is the amino acid sequence that determines the protein's chemical properties and function. There is more than one hemoglobin gene. In humans, hemoglobin A is coded for by the genes HbA1, HbA2, and HbB. The amino acid sequences of the globin proteins in hemoglobins usually differ between species. These differences grow with evolutionary distance between species. For example, the most common hemoglobin sequences in humans and chimpanzees are nearly identical, differing by only one amino acid in both the alpha and the beta globin protein chains. These differences grow larger between less closely related species. Even within a species, different variants of hemoglobin always exist, although one sequence is usually a most common one in each species. Mutations in the genes for the hemoglobin protein in a species result in hemoglobin variants. Many of these mutant forms of hemoglobin cause no disease. Some of these mutant forms of hemoglobin, however, cause a group of hereditary diseases termed the hemoglobinopathies. The best-known hemoglobinopathy is sickle cell disease, which was the first human disease whose mechanism was understood at the molecular level. A separate set of diseases called thalassemias involves underproduction of normal and sometimes abnormal hemoglobins through problems and mutations in globin gene regulation. All these diseases produce anemia. Variations in hemoglobin amino acid sequences, as with other proteins, may be adaptive. For example, hemoglobin has been found to adapt in different ways to high altitudes. Organisms living at high elevations experience lower partial pressures of oxygen compared to those at sea level. This presents a challenge to the organisms that inhabit such environments because hemoglobin, which normally binds oxygen at high partial pressures of oxygen, must be able to bind oxygen when it is present at a lower pressure. Different organisms have adapted to such a challenge. For example, recent studies have suggested genetic variants in deer mice that help explain how deer mice that live in the mountains are able to survive in the thin air that accompanies high altitudes. A researcher from the University of Nebraska-Lincoln found mutations in 4 different genes that can account for differences between deer mice that live in lowland prairies versus the mountains. After examining wild mice captured from both highlands and lowlands, it was found that the genes of the 2 breeds are virtually identical except for those that govern the oxygen-carrying capacity of their hemoglobin. The genetic difference enables highland mice to make more efficient use of their oxygen, since less is available at higher altitudes, such as those in the mountains. Mammoth hemoglobin featured mutations that allowed for oxygen delivery at lower temperatures, thus enabling mammoths to migrate to higher latitudes during the Pleistocene. This was also found in hummingbirds that inhabit the Andes. Hummingbirds already expend a lot of energy and thus have high oxygen demands, and yet Andean hummingbirds have been found to thrive in high altitudes. Non-synonymous mutations in the hemoglobin gene of multiple species living at high elevations have caused the protein to have less of an affinity for inositol hexaphosphate, a molecule found in birds that has a similar role as 2,3-BPG in humans. This results in the ability to bind oxygen in lower partial pressures. Hemoglobin adaptation extends to humans as well. Studies have found that a small number of native Tibetan women have a genotype which codes for hemoglobin to be greater saturated with oxygen. Natural selection seems to be the main force working on this gene because mortality rate of offspring is significantly lower for women with higher hemoglobin oxygen affinity when compared to the mortality rate of offspring from women with low hemoglobin oxygen affinity. While the exact genotype and mechanism by which this occurs is not yet clear, selection is acting on these women's ability to bind oxygen in low partial pressures, which overall allows them to better sustain crucial metabolic processes. Synthesis. Hemoglobin is synthesized in a complex series of steps. The heme part is synthesized in a series of steps in the mitochondria and the cytosol of immature red blood cells, while the globin protein parts are synthesized by ribosomes in the cytosol. Production of HB continues in the cell throughout its early development from the proerythroblast to the reticulocyte in the bone marrow. At this point, the nucleus is lost in mammalian red blood cells, but not in birds and many other species. Even after the loss of the nucleus in mammals, residual ribosomal RNA allows further synthesis of HB until the reticulocyte loses its RNA soon after entering the vasculature. Structure. Hemoglobin has a quaternary structure characteristic of many multi-subunit globular proteins. Most of the amino acids in hemoglobin form alpha helices, connected by short non-helical segments. Hydrogen bonds stabilize the helical sections inside this protein, causing attractions within the molecule, folding each polypeptide chain into a specific shape. Hemoglobin's quaternary structure comes from its 4 subunits in roughly a tetrahedral arrangement. In most vertebrates, the hemoglobin molecule is an assembly of 4 globular protein subunits. Each subunit is composed of a protein chain tightly associated with a non-protein heme group. Each protein chain arranges into a set of alpha helix structural segments connected together in a globin fold arrangement, so called because this arrangement is the same folding motif used in other hemoglobin proteins such as myoglobin. This folding pattern contains a pocket that strongly binds the heme group. A heme group consists of an ion held in a heterocyclic ring, known as a porphyrin. This porphyrin ring consists of 4 pyrrole molecules cyclically linked together with the ion bound in the center. The ion, which is the site of oxygen binding, coordinates with the 4 nitrogens in the center of the ring, which all lie in one plane. The ion is bound strongly to the globular protein via the imidazole ring of F8 histidine residue below the porphyrin ring. A 6th position can reversibly bind oxygen by a coordinate covalent bond, completing the octahedral group of 6 ligands. Oxygen binds in an end-on bent geometry where one oxygen atom binds Fe and the other protrudes at an angle. When oxygen is not bound, a very weakly bonded water molecule fills the site, forming a distorted octahedron. Even though carbon dioxide is carried by hemoglobin, it does not compete with oxygen for the ion binding positions but is bound to the protein chains of the structure. The ion may be either in the Fe2+ or in the Fe3+ state, but ferrohemoglobin cannot bind oxygen. In binding, oxygen temporarily and reversibly oxidizes to while oxygen temporarily turns into superoxide. Thus iron must exist in the +2 oxidation state to bind oxygen. If superoxide ion associated to Fe3+ is protonated, the hemoglobin ion will remain oxidized and incapable of binding oxygen. In such cases, the enzyme methemoglobin reductase will be able to eventually reactivate methemoglobin by reducing the ion center. In adult humans, the most common hemoglobin type is a tetramer called hemoglobin A, consisting of 2 alpha and 2 beta subunits non-covalently bound, each made of 141 and 146 amino acid residues, respectively. This is denoted as alpha-2 beta-2. The subunits are structurally similar and about the same size. Each subunit has a molecular weight of about 16,000 daltons, for a total molecular weight of the tetramer of about 64,000 daltons. Thus, 1 gram DL equals 0.1551 mmol/L. Hemoglobin A is the most intensively studied of the hemoglobin molecules. In human infants, the hemoglobin molecule is made up of 2 alpha chains and 2 gamma chains. The gamma chains are gradually replaced by beta chains as the infant grows. The 4 polypeptide chains are bound to each other by salt bridges, hydrogen bonds, and the hydrophobic effect. Oxygen saturation. In general, hemoglobin can be saturated with oxygen molecules, or desaturated with oxygen molecules. Oxyhemoglobin. Oxyhemoglobin is formed during physiological respiration when oxygen binds to the heme component of the protein hemoglobin in red blood cells. This process occurs in the pulmonary capillaries adjacent to the alveoli of the lungs. The oxygen then travels through the bloodstream to be dropped off at cells where it is utilized as a terminal electron acceptor in the production of ATP by the process of oxidative phosphorylation. It does not, however, help to counteract a decrease in blood pH. Ventilation, or breathing, may reverse this condition by removal of carbon dioxide, thus causing a shift up in pH. Hemoglobin exists in two forms, a taut form and a relaxed form. Various factors such as low pH, high CO2 and high 2,3-BPG at the level of the tissues favor the taut form, which has low oxygen affinity and releases oxygen in the tissues. Conversely, a high pH, low CO2, or low 2,3-BPG favors the relaxed form, which can better bind oxygen. The partial pressure of the system also affects O2 affinity where, at high partial pressures of oxygen, the relaxed state is favored. Inversely, at low partial pressures, the tense state is favored. Additionally, the binding of oxygen to the iron 2 heme pulls the iron into the plane of the porphyrin ring, causing a slight conformational shift. The shift encourages oxygen to bind to the 3 remaining hemes within hemoglobin. Deoxygenated hemoglobin. Deoxygenated hemoglobin is the form of hemoglobin without the bound oxygen. The absorption spectra of oxyhemoglobin and deoxyhemoglobin differ. Deoxyhemoglobin has significantly lower absorption of the 660 nanometers wavelength than deoxyhemoglobin, while at 940 nanometers its absorption is slightly higher. This difference is used for measurement of the amount of oxygen in patients' blood by an instrument called pulse oximeter. This difference also accounts for the presentation of cyanosis, the blue to purplish color that tissues develop during hypoxia. Evolution of vertebrate hemoglobin. Scientists agree that the event that separated myoglobin from hemoglobin occurred after lampreys diverged from jawed vertebrates. This separation of myoglobin and hemoglobin allowed for the different functions of the two molecules to arise and develop. Myoglobin has more to do with oxygen storage while hemoglobin is tasked with oxygen transport. The alpha and beta-like globin genes encode the individual subunits of the protein. The predecessors of these genes arose through another duplication event also after the gnathosome common ancestor derived from jawless fish, approximately 450 to 500 million years ago. The development of alpha and beta genes created the potential for hemoglobin to be composed of multiple subunits, a physical composition central to hemoglobin's ability to transport oxygen. Having multiple subunits contributes to hemoglobin's ability to bind oxygen cooperatively as well as be regulated allosterically. Subsequently, the alpha gene also underwent a duplication event to form the HbA1 and HbA2 genes. These further duplications and divergences have created a diverse range of alpha and beta-like globin genes that are regulated so that certain forms occur at different stages of development.